pore namija. Hello everyone. Good evening. Welcome to our institution of Global Professional Free webinar. Thank you all for joining and stay with us till the end. My lovely audience, today I am your host. My name is Yarni Sakhtar and I am from Bangladesh. I feel very proud to host this webinar. I feel privileged to host this webinar. I am associated with IGP as a global member. It's my delight to welcome all of you is our free webinar. Thank you all for joining us and stay with us till the end. And don't forget to tag, share, and mention your friends. Today our webinar number is 1050. And our webinar topic is recent trends in application of artificial intelligence. And our speaker name is Dr. Harish Chandra. He is from India. Let's welcome our speaker.
And can I start the presentation, ma'am?
Hello, sir. Uh,
Yes, ma'am. Hi.
Good evening, sir.
Hmm.
Sir, I
Okay. Good evening.
welcome in our webinar. First, you have to give some
Okay.
description about yourself. Then you can start your presentation.
Okay, ma'am. Well, a small introduction about myself. Dr. Harish Chandra Anantram completed my bachelor's and master's in bioinformatics, completed my doctoral degree in bioengineering. I have applied some machine learning techniques to identify patterns of microRNAs from the transcriptome data. And some of the microRNAs are even in clinical trials also. And I hold a few good quality publications and patents. And right now, I'm in the process of identifying novel biomarkers for autoimmune disorders. So, with respect to today's topic, application of artificial intelligence in various fields. First, I'll be discussing about the application in my field, and I'll be discussing with the application in other relative fields also. And to go ahead, let me give you a basic introduction. Well, in case of bioinformatics, why the concept came because diverse biological application require a diverse graph theory. Because why graph theory came into picture? Because now people talk about the systems biology as a whole, not a specific gene level or a mutation level. When it comes to systems biology, each and every component you have in the environment have a direct or indirect connection to the component of every species. Based on the logic only, we have molecular clock hypothesis and circadian rhythms. So it's a single reason that no single graph topic can cover the entire biology. You need multiple graphs and finding the minor differences between the multiple short hubs in the graph, and only those concepts will be giving you a detailed feature. Even there are more opportunities for the people who work on the combinatorial optimization part. Next is systems biology. In terms of computational analysis, we call it network biology. In case of the evolutionary as well as the species based phylogenetic related study, you use the term systems biology. So, the basics of network biology involves the gene regulatory networks because gene is regulated either by a microRNA or a long non-coding RNA. Now people talk about circular RNAs. And then we do have the protein interaction networks because protein interaction networks will be from the coded part of the gene and the regulatory networks will be from the non-coded part. They cover the whole of systems biology. And we do have other relationships in biology representing graphs as networks. For example, microbiome. in the past 2 years, there are a lot of papers being published in microbiome, and there are a lot of patentable products which are being sold in the area of microbiome. And then what problem we get computationally is the recurring problem, because most of the data in graphs are in terms of the cluster. So the problem is finding a proper click or the variance of a minimal click, that is called as the hub in terms of biology. So finding that becomes a little tough process. For that, we use the recurring functions, and we do have a lot of variance of the graph in isomorphism in the network motif. What do you mean by isomorphism? In the sense, a single gene can code for multiple proteins, and in the same way a single protein can be connected to multiple protein. And proteins, as we know, they do not work in isolation. They work as a part of compatibility complex, and for each and every orientation you have the conformational variations that is resulting in the structural variation also. That concept we call it as the isoform. And then we do have the molecular pathway based problems also. So pathway reconstruction problem is one of the vital problems with every group is working on. Why? Because they need a random graph theory to test the level of significance. The next is, in terms of computational study, we call it as a click problems. Click problem is an outcome of recurrence in clustering applications, but true clicks are computationally hard to find. So the people suggest various computational models through graph theory, that is trackable, biologically meaningful alternatives with respect to clicks. As an example, we can see the maximum density subgraph and the extreme sets in a graph. So we'll go a little deeper into the subgraph density. What happens is, if a given graph contains a set of nodes and edges, and each node is connected to the other node by a single or even multiple edges in some rare cases. So the maximum density subgraph gives the maximum space for the elongation of the stretch with respect to the modulus. For each and every node, the calculation is done for the maximum density subgraph. Then comes the maximum density subgraph with respect to polynomial time. That is the space and time complexity analysis, how it is taken care of is also being dealt. So comes the maximum click with different properties. So we take some extreme sets. So in case of edge weight undirected graph, because in our biological systems, we always don't get direct graph like the example problems we deal in schools. We mostly get the undirected graph because we can't specify a protein should code only to these many proteins. It can code to many proteins also. And similarly, the gene regulation also. So the undirected graph we are taking, so we compute the total weight and the edges associated with the graph, and all extreme sets in a graph can be found in a polynomial time. And next is the pipeline development. Pipeline development is a core area which many computational people work along with biologists to give some meaning from the various biological experiments generated by the high throughput techniques. What they do is, so there is a need for more sophisticated applications using the random graph theory to study biological networks. And indeed, they establish even the basic null model with the minimal set to start. A null model is not always where there is nothing. A minimal set, no, in a graph is called as a null model, because in a minimal set you can't compute the maximum centrality measures as well as the degree. So this significance is taken care by the subgraph, and various patterns are found in the biological networks. The next part is to distinguish the observed patterns in the subgraph. Observed patterns can be, suppose, for an example, if you working on the Krebs cycle, the carboxyl metabolism is one of the observed pattern. And related to that, we can also work on the biologically appropriate model, because there are a lot of computational models which are in use now, but very few are biologically relevant. The one model which is biologically relevant is the ontology model. They cover the biological process, molecular function, as well as the cellular component. But the cellular component is not given much preference because proteins do not stay in one particular cellular component. They keep moving to the other components also. And with respect to the outline of the graph theory, in application to genomics, the Euler's path is being applied to the sequence analysis, and the tanglegrams are being applied to the co-evolution based analysis. The network design is incorporated for making the computer to understand the multiple sequence alignment, and the haplotyping by perfect phylogeny is based on the graph realization. Then the polygenetic networks is also a very good topic now for research. Polygenetic networks, they use incompatibility graph here, and lot of epigenetic studies are being done in polygenetic networks. And next is the galleries. They use recombination networks to understand the galleries. So, with respect to any computational algorithms, the initialization comes first. The Euler's path in sequencing, the sequence assembly is taken care first, and then the tanglegrams are used for testing the coevolution, and then they build the tree design. And this is the interplay between the trees and the string, the design algorithm we use. So, in case of the sequencing problems, what we generally use is we take a DNA molecule, we split it in terms of a k-mer. Since we know the triplet codon model, most cases for the linear sequence, the k-mer starts with 3, and for circular RNAs, it can go up to 5 and 6 also to fit the dimensional of the circle. Then we use the k-mers to which we want to determine the actual set of sequence for the initial assembly, and then we determine the possible set of sequence based on the alphabet size, and then we create a novel approach to build a Eulerian diagram based on the k minus 1 mass, because this is to avoid the space and time complexity. If you take the general k as a whole, the time will exceed, so k minus 1 model is being incorporated. So for the general k, what we use is the k minus 1 mer is converted to a k mer, and then the k minus 1 mer is converted to k minus 2 and k minus 3 also, but accommodated with the additional notations in various stages. And then for k equals 3, we can see the graph will have one node for each of the two mers, observing the 3 mer, and there is a direct edge from the node 2 mer from the concurrent point. So xy is here, then yz will be, and the cycle is being repeated. So the sequence assembly is being carried out from these cycles. This is a small example, but in general, sequencing machine can read only 10,000 characters, but we have billion characters in the genome. This process has to be completed for the whole genome assembly. In case of eukaryotes, it's not an easy task. Then the point is, every Eulerian path in the graph specifies a sequence k-mer match to the given data, and conversely, every sequence whose k-mer matches the specifier follows an additional Eulerian in the graph, and the path gives you the candidate sequence for the unknown original sequence. And there are a lot of algorithms which do exist for efficiently finding the Eulerian paths with the counter number, and they give some unique models with respect to the candidate sequence. And comparing to this approach, our efforts represent the set of candidates by a graph with a Hamiltonian path, and each node represents an observed k-mer, but not a k minus 1. Though we give k minus 1 for simplicity, in the computer which takes the k and putting the k minus 1 to solve the computational optimization. So we need to make finer distincts on the basis of Euler path. In general, there can be many Eulerian paths in the graph, and we may want an additional criteria to distinguish which one is good when we compare with the other one. So we consider different translation models. That is, from the machines we get only the signal, and we have a software to translate the signal into the sequence. And with respect to the subpath separation, those two sequences get aligned, and the length between each read also can be calculated. Then the value of Eulerian path P with nh is the sum of n minus 1 value with respect to the length difference between the two subpath in P. The problem is to find a Eulerian path with minimum value. We have some reasonable approximations to work on this, but a simpler case can be solved by a basic optimal solution in a polynomial type. So we'll start from a binary alphabet and how the arbitrary. Suppose you have a KMA4, so n minus 1 it takes the 3 combination. You have a root and 4 relative species from the root, and how the molecule fits the alignment will be seen now. Because when it comes to computational programs, if you type 2 or 3 commands, you will get the complete assembly. But how the system works, I'll explain. So at any node, there are two possible ways for a Euler path to pass through. You can see either way. And then you can have a crossing between these two. Because in genetic algorithm, we talk about crossover, and Mendelian genetics also gave a detailed description for the term crossover. And we are restarting to the optimal Euler problem. We are giving a Eulerian graph where the in degree as well as the out degree. Degree is the number of connections the central node has with the other node, and how far and how come they are minimal distance, and how far and how come they are far distance gives you the in degree and the out degree. And for each given value, the turning pair and the value for crossing pair is being calculated. Then we choose the tuning and crossing pair at various nodes to maximize the total value of the choices. And all our assumptions are subjected to the requirement of creating a minimum meaningful choices to create a proper Eulerian path in the graph. And the solution involves, even now a polynomial time is required for getting the complete solution. The set of choices given by the Eulerian path have a matroidal structure, that is a massive structure. So they use some greedy algorithms, that is give the output faster based on the parameters to find the optimal Euler path, and the more direct algorithm based on the minimum spanning tree can also solve the problem. So we'll see how the matroid structure looks. At every node V, the H pair crossing or turning has the lowest value is called as the low pair, and the other which has a high value is the opposite of low, and the difference in values will give a loss at B, and the subset of S node is independent of the subset of the Euler's path, and a lower pair is chosen. So the definition says the family of independent sets from a matroid can also be found by a greedy algorithm, but the independent functions has to be minimum to give the optimal path. And then we have the tanglegram. In tanglegram, a pair of trees are drawn in a plane with no crossing edges with the same label leaf set. The leaves of one tree are displayed on a line, and the leaves of other are displayed parallel. So a straight line connecting each leaf in one line to the leaf with the same label gives you the direction to the other tree. So the number of crossing lines is a measure of the similarity between the trees. So let's see how the multiple sequence alignment can be studied. So here we have the interplay between the basic sequence and the trees, and we explore a network design based approximation. And further, the study can be extended to population genomics. Right now, around 14 to 15 people, the complete genome sequence is done. because previously the report where they say the completion of human genome project is only the exome sequence, not the whole genome. As far as I know, with respect to current literature, for only 14 people it's completed, and people are trying to extend it for 10,000 genomes, and very few places they can work it out, the 10,000 genome project. Now we want to sequence many individual in a population is our task, and we require to correlate the similarities and differences in the sequence with the genetic traits. So we can determine whether the person is disease or he is susceptible to disease based on the nature of sequence. Presently, we can't sequence large individuals due to various constraints with respect to the equipment, with respect to the availability of
Sir,
additional analysis. Yeah.
I think your slide is not moving.
Okay. 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 Now it's moving, right? Yeah.
Yes, yes, now it's moving.
Yeah. Okay. I'll come to. Then we'll come to slide number 23. Okay. Next, when we analyze the single nucleotide polymorphism, that is the specific site in the genome where two different nucleotides apply with sufficient different frequencies in the population, say 5% of the frequency variation. SNP maps will give you a compiled density for a site, like one site per thousand sequence can be found. That is the ratio they have calculated for the SNPs. And to give a classical example of the SNP, in most of the textbook you come across the sickle cell anemia disease, because in the sixth position it has to be the glutamine amino acid that is will be replaced by valent for the people with sickle cell. So the RBCs will be in sickle shape. So that particular mutation changes the entire structure. And there are two things to note here. The people who have sickle cell anemia will not get malaria, but they have a lot of tough time with this disease itself. Why I'm saying they will not get malaria? Because malarial vectors cannot insert their proteins to do the differential action. Okay. So with respect to the SNP data, it's easy to collect in larger population, and it's a little cheaper also compared to other markers. And then we'll be discussing how the HapMap project is connected. Because the term HapMap refers to the haplotype mapping, NIH have funded this project to find the common SNP haplotypes in the human population. And with respect to the association mapping, HapMap tried to associate the genetically influenced disease with specific SNP haplotypes to find the region near the casual mutations. So the key logic is to perform an associational mapping with historical recombination in the population. Nature has done experiments, and now we try to make sense out of the results, how the haplotype varies. So now we'll be studying the perfect phylogeny haplotyping via graph realization. When we try to differentiate the genotypes and the haplotype, as we know clearly that each individual have two copy of chromosome. At each site, each chromosome has one or two alleles, and in computationally they can be represented by 0 and 1. It is motivated and differentiated by SNPs. So the two haplotypes from one individual merges to the two haplotypes to the related person, and the genotype of the individual will be slightly different from the haplotype. So how it is different? Because a single genotype can have a relatively variable haplotype. Like that, the difference can be understood. So you can see the difference of the two haplotypes with respect to one genotype. A combination of these two haplotype can result in a genotype. It's not always straight, because 0 and 1, it has to be giving you the answer 1, but it's giving 2. So the additional insert is added here. So 1 and 1 supposed to be 2, but it is 1. So 1 point mutation is here. So likewise, a lot of differences need to be captured. For capturing these differences only, we use the AI and ML models. So computationally, how this haplotype problem is studied, because if given a n genotype, we need to determine the number of haplotype pairs, and how it can be differentiated from a genotype, and how the haplotype can perform without affecting the genetic model, because the core model will not be different, only the haplotype variants will be different, and those things, the computational problems we try to solve, require to address these challenges. So, in case of the biological problems, in case of disease association studies, the haplotype data is more valuable than the genotype data. But haplotype data is very hard to collect, and the genotype data is easy to collect. So, based on these relations, we can predict as well as do an experimental assay for confirmation. So then the perfect phylogeny model for SNP sequence is being analyzed. You have 5 sites in the initial ancestor where there is no much additional functions in terms of the natural mutations for development or certain other missense and non-synonymous or synonymous mutation. So in that case, you start only with the 0, and you have various child from the ancestor, and you represent with the relative number, and how the phylogenetic analysis works with respect to the root and the tree. Oh, sorry, the root and the child, and tree is the general structure. Based on the tree, it can be either a dendrogram, that is without proper distance from child to the root, or a phylogram with proper distance from the child to the root. So, when we can set a number of sequences between a derived child to understand the perfect phylogeny model, the classic NASE algorithm arranges sequences in a matrix and no duplicate column is allowed. So the sequence can generate a unique perfect phylogeny, and if only no column contains all 4 pairs. Because if column contains all 4 pairs, then people will work on the hidden Markov models to find the hindrances. When it comes to hidden Markov models, you have 3 states in the model: match, insert, and delete, and you have a transition probability matrix between these states to analyze the process. So the binary characters associated with each column allows the tree to for the entire set of analysis. So there are m dimension for the relative data m exists for a perfect phylogeny in the ONM time. That is the higher order complexity, and a person called Dan Gusfield have worked on the graph theory part of it, and his recent book is also on applying the integer linear programming systems biology. So we'll be using those classical theorems to apply in biological problems and solve those problems and identify the patterns which make sense. So in case of the perfect phylogeny model, obviously there has to be an assumption for the phylogeny because the root cannot be completely determined either by experimental or by the simulation model. So the one which is relative can be taken as a root with a further distance. The nearest distance cannot be a root. A far distance only can be taken as a root based on the relative characters. So one edge in the haplotype is created by accumulating the challenges for the leaf where the haplotype is displayed. In other words, we can say the extant haplotypes evolve along a perfect phylogeny with all zero root. As I told you, the root will not undergo any possible mutations, so the number will be zero only. And then, with respect to the justification, the haplotype blocks give you the rare recombination as well as the base problems to develop your solution in a modified aspect, and you can solve biological complexity to a certain extent. So the perfect phylogeny haplotypes can be determined by a small matrix with respect to the genotype. Yes, finding the set of haplotypes to fit the phylogeny. The haplotype pair explains the genotype. If the merge of the haplotype is created, so the matrix of the merge can have 0 and 1 in one point and 1 0. The other point will explain you the similarities and differences. And then the PPH problem, what it says is that if you have 2 and 2, it has to be either 2 or 0, but you have 1 and 0 model here, then you can say there is a change here, whether it's a mutation or SNP, we don't know exactly. And this is the problem which many people use many methods to try and explore the distinctive patterns in the phylogeny. And the problem can further be extended to graph for getting a simple solution in less number of steps. So the alternative explanation in computational point is no tree is possible to understand this model, but biologically there are systems with this model and we need advanced algorithms to explain it. So the efficient solution is reduction of the graph realization problem, build a Wenger fishable graph with respect to ON model compared to the basic NM, and then the reduction of graph size based on the turtle graph realization. And these are from the Gusfield only, and he's the person who has done extensive work in applying graph analytics to biological sequence analysis, and a lot of recent models he's developing. In his book, he has given around 60 exercises for practice. And then we have the Berkeley approximation with respect to the linear solutions. Then the reduction approach. Here we take the polynomial time as such instead of the nodes in the graph, and we conceptually simplify the problem from higher level to an implementation level, and most extendable problems are nearly the linear time but not exactly the linear time, because no biological model can be understood by linear machine learning algorithms. You require a lot of non-linear machine learning algorithms to understand the problem. So with respect to the case of ones, for any row I in S, the set of ones entry is a row I specific to the exact set of mutations on the path from the root with the least common ancestor. So that only the labeled leaf I in every perfect phylogeny will be equivalent to the S. So in order to get those entry specifically with one, the path also should be same in every perfect phylogeny. So we determine the leaf counting here. And then in any column C for the two internal nodes to get the value to the total number of the leaves in the mutation should have a perfect phylogeny for the S. You can see the matrix here. You have seven relative roots and seven relative childs, and how far and how come they are related will be explained by this model. So the simplest conclusion is the order in red is the mutation, and together we have is the susceptible one, which may get mutated in a longer time. So the basic idea is for a perfect phylogeny, for every yes, no, it should have the row I and the column C with the equidistance or a lesser distance. If it is very far distance, then the perfection in phylogeny will be a question mark. We need additional algorithms to explain it. Maybe the maximum parsimony or likelihood algorithm can explain this process, but other basic algorithms with respect to arithmetic progression, it's tough to explain. So the path between two leaves labeled must contain the edge with mutation C. Further, every mutation on the path between these two leaves must be from the same column. That is also an additional aspect. So from the row data to the tree constraint, you can see there can be additional edges also. In protein-protein interaction networks also, you might have a basic networks based on the gene level study. Now, a lot of networks are being updated based on the genome-wide association studies and the whole genome projects. So, with respect to the basic theory, we create a directory and we use integers to represent the root and the leaf, and we have a subgraph because if root is a main graph with two child, the subgraph can be multiple childs, and the second child will be the root for the third child, and third child will be the root for the fourth child. So to represent mathematically, the Rn will be the integer and P will be the subset, and the tree T associated with the relative integer n gives you the family of P1 to T, and the graph realization problem generalizes the constructive model to understand the oneness path in T. This is similar to the traveler salesman problem also. And then more generalization for each subset specifies the fundamental cycle in a known graph. So the tree realization example gives the first path as 158 as one model, and the second path is only 24, and for the third path it's 1 2 5 and 6 little extension, and for the fourth path it's only 3 6 and 8, but for the fifth path you have additional extension. And this is how the path finding algorithm works. So reducing the PPH to graph realization, we know at some instant the PPH problem is created by the various number of path set, and each and every path set can result in a graph realization problem that is leading to another PPH instant. So how we need to encode the minimal subgraphs to give you the whole picture of the complexity in biological networks and how to glue them together at the root. That is the challenge which many people are working on, and there are some alternative solutions to address the challenge also. So from the raw data to the tree constraint, how the adjacent spots are worked out. So we encode a red-blue directed path from these three child, and the root is kept as u for the two path. For the other path, it's only the child, so the root is not being taken. So you have a glue-based directed path. Now we add an additional path to the row I and get the model, and the resulting path gives you the encoding for everything that is known about the row I in the input, and the family of the set of lesser path gives you the alternative input to the graph realization problem, and every solution to the graph realization problem specifies a solution relative to the PPH problem in an opposite way. So in this way we can see the relation and develop pipelines accordingly. So the implicit representation to all solution was started by Whitney in 30. He gave the reconnected graph model, and he also proposed the uniqueness and non-uniqueness between graph and the twisted models. And the tree realization came to give you the connectivity model with respect to the shortest path. And then the phylogenetic networks along with recombination analysis were understood. So when a set of sequence is derived from a phylogeny, the classic NSAC example is only 0, 0, 0, 1, 1, 0, and 1, 1, but this is not always feasible. We have some incompatible sites also. The best example for this incompatible sites is that in RNA you have the opal hypothesis, because non Watson pairs also can be aligned in RNA, because it's not D-deoxyribosugar, it's only a ribo sugar, and in DNA it's only the 3 prime and 5 prime, and not mandatorily feasible in RNA. So this that is the example you can use to understand the incompatible sites. And a richer model will give you the relative aspects to understand the incompatibility. So in case of the sequence recombination, as we know about having maximum from one parent and minimum from the other parent to give you the child, you take 4 here and 1 here on the base of homologous recombination, you get a relative number, and then you use networks to understand the recombination of the basic model. Here they have mentioned it as ARG, but you can mention any relative codon with respect to the model. And now we will be seeing the association mapping of complex diseases by using the recombination. So in case of association mapping, since SNPs are easily available and require a minimal experimental cost, they are taken, and then we do a simplest map for unobservable mutations also associated with mental disease. You have a complexity network here. This network can be a small social network also, where you have multiple friends and you have a different level of connection with each friend. Like that also the network can be made. So in case of mapping disease with the inferred arguments from the SNP data, those arguments can be given as a result of reconstruction in the evolutionary analysis. So first we need to clean the mathematical algorithm results, and we give some practical composition to the constraint, and we supply some biological inputs to define the accuracy of the model to be feasible, and we extend the model to the gene conversion matrix, and we give the minimal set of optimization to understand the process of the complete recombination model. So if we don't have a tree data structure, then by using which data structure we are going to use it, we have a tree like data structure. The only difference between the tree and the graph is in tree, in as basic cyclic model is required, but in graph it's not always required to be cyclic. So a tree like network can have similarly the root is unknown and the child and the covariance with respect to the distance is given, and the recombination cycles are understood from the phylogenetic network with two path, that is one is front and one is back, and eventually you can have the path association in the specified matrix. So each and every cycle specifies a minimum of two paths to be connected to understand the recombination cycle. And you have a Galtree model, a phylogenetic networks where no recombination cycles share an edge as the Galtree. A cycle is a simple Galtree, and every node from the cycle is a gal. Question is if the m set of rows cannot be generated on a true tree, can it be generated from the gal tree? This is how the gal tree looks, slightly different from the tree, and based on this only the algorithm is created. So that only most of the machine learning algorithms are biologically inspired. So the results we obtain from gal tree is the proven set of solutions in the polynomial time, and we determine the algorithm is very specific in the gal tree. And then we also have a theorem that is supporting the gal tree to fit the recombination model. And the theorem is relatively simple. If M can be derived from a gal tree, then the gal tree is really unique. From this only the molecular clock hypothesis and circadian rhythms were further studied. So when it comes to uniqueness, the number of arrangements on the sites on a gal happens if it has two sites, and then the gal can be extended for more than two sites based on the number of arrangements. If the gal has four more sites, which of these two sites are related in recombination gives you an exact recombination point, and the one that is on the other side gives you the unique point. So therefore, the invariance is understood. And then by using all these ideas, what are all results we can obtain? We can obtain the incompatible sites. That is a pair of sites M that fails the four game set in a basic test to get the incompatibility. And site that is not having the complete pair is compatible because you can get a result by using only one pair of chromosomal data to understand recombination, but that is not the way to do it. You need to consider both pair from both parents to get the complete data of the child, so that only the incompatibility occurs. Because when you get the details of both pair, obviously there won't be synchronization, and for dealing the non-synchronous data there are separate algorithms and additional work is required to be done. So you have an incompatibility graph. Here the advantage of using the main tool is we represent a pairwise incompatibility in an incompatible graph. That is the understanding we need to have. So the connected components of GM are very informative. So the theory says non-trivial connections, that is not to complete connections in the components in lower bound, have maximum number of recombinations needed in any network. And then it can be extended as a single derivative of a gal tree to get all incompatible gal tree outcomes with a single connection itself. So the gal tree gives you the number of recombination associated with the major component, where G is the major gal root and M is the subset, and hence the minimal possibility to find a logistic recombination of phylogenetic networks is determined by the M itself, because no need to explicitly calculate the G. And you have some incompatibilities here, because in all these areas you have four sets, but here you have only three possibilities. Since you don't have a root, you take this 4 as a root, and then 1 and 3 as this module, and 2 and 3 the straight graph can be taken. You can see a direct straight connection, no? So that comes the incompatibility. So the graph theory is associated with the necessary condition for the gal tree. If M can be generated on a gal tree, then the incompatibility of the graph must be either a bipartite or a biconvex graph associated with the other structural properties, or if a conflict graph comes, then it can be detected and further exploited. Then the Galtree haplotyping is studied. Given a genotypic matrix, if there are no PPH solutions for the graph G, then there is a haplotyping point H for the same graph G. Then the H can be derived on the Galtree. So a different necessary condition for satisfying one Galtree gives the existing set of sequences such that every pair of incompatible sites PQA, the single PNQ gives the basic states appearing in all sequences in the total set of sequences and does not appear in any sequence outside S. That is the condition we need to specify for the computer to understand. Then we must specify the number x such that the probability of x for each incompatible pair P and Q. Because in basic probability matrix, 1 is the maximum, no, P plus Q based on the associative property. So you have an example here with multiple sets. So you can take a single sequence combining two characters A and D is associated with the graph to perform haplotyping. So coming to the full circle, back to genotypes, when we can get a set of genotypes to be explained by a set of haplotypes derived from the Galtree rather than from one perfect phylogenic model, the NASC can be translated into ILP, that is the integrated linear programming, and the minimal ILP solution will identify each pairs of incompatibility. So for the one gal problem, if we use the ILP formulation to solve it efficiently, since we have 20 amino acids, you have 200 rows and 40 sites or for each amino acid, 5 different confirmations are allowed, so 40 sites. A second is given in a minute. So when we try to minimize the recombinations in unconstrained networks, we try to use Galtree with respect to the minimization algorithms, and they do have biological meaning also because most of the drugs start their interaction in a minimized form only, so that only energy minimization gives a lot of relevance in studying biological systems. So the minimization is obviously a NP hard problem. So to solve the problem, we have a smaller data initially at exponential time with all associated methods, and then we efficiently compute the lower and upper bounds in the relative recombinations, and then we apply these methods to address specific biological and biotechnological related questions. So in the decomposition theorem, we minimize the NP hot problem and we want to break up the problem into sub problems and we solve it separately to get combinatorial solutions. And the incompatibility graph can be associated with the subsequent matrix also. The main tool here is we represent a pairwise incompatibility in the graph based associative matrix. The connected graphs are very informative. Non-connected graph also, they do have a meaning, but you should be smart enough to identify the level of connection and make it connected. So in case of the recombination cycle, the phylogenetic network with node X can be tracked with the minimum number of path backward from X, then the path will be eventually connected. The cycle specifies these two paths as the recombination cycle. The minimal set of interesting cycles form a blob also, that is the biconnected network. If the direction of each edge is complete, a blob or biconnected network can be formed. So since the gal tree is very complex, people try to study the blob trees. Connecting each blob in a network result in a directed network and a rooted tree. Otherwise, one of the blob, which was not maximal, can have a simple but give you a key insight. So every phylogenetic network can be viewed as a directed tree blob. So a blobbed tree is created instead of a gal tree. So the blobs are non-tree like pods, but they still have an association in the network. So you can see the network in terms of blob. A network where a blob is single cycle gives you a gal tree. So you can see the difference as well as the association. Tangled networks inside a blob. A simple observation is in any network n for m, all sites from the same or connected by the component g of m must appear together in a single blob. So the decomposition theorem gives you the sequence m. There is a phylogenetic network that derives m where a blob is allowed once, and then the site in one non-trivial connected component gm gives the relative connections for the site when placing the edges together in other block. So the fully decomposed network gives the finest decomposition possible. And then network of input m with one single blob is shown with respect to other notations. These are the outcomes of the signal. Like you can see for the various child, various relative signals are being created. And the fully decomposed network M is also shown, just like the Galtry network. Moreover, the backbone tree is invariant over the fully decomposed networks, and the network is determinantal in polynomial time. So we can find a network for the matrix M by solving the recombination minimization problem for each connected component of the graph with the matrix separately, and then connect them in subnetworks to find the invariance. Algorithmically, we can find the tree part of the block tree in a easier fashion, and we can determine the sequence labeling from exterior node by using blob, and we can determine a good structure on the basis of blob so that we can generate a sequence. It's easy to test whether the exterior sequence is present or not present on the blob, which is generated by the base of a single recombination. Then the original Galtry problem is now just a problem of testing one single crossover recombination sufficient with each block. Then they can be solved successfully by removing the external sequence and testing the remaining sequence can be generated on a perfect phylogeny to correct the graph. So the sufficient conditions are, if we prove several useful sufficient conditions where there is a fully decomposed network that minimizes the number of recombination over a possible network. In the deep result, let n be a phylogenetic network for input m and L be a set of sequences for the label nodes n. Let G of L be the incompatible graph for L. If G of L and G of M have the same number of connected components, then there is a fully decomposed network for M with the same number of recombinations M. And a fully decomposed network exists that minimize the number of recombination unless every optimal network uses some recombination node labeled by the sequence. It's tough to find the matrix M, and then the addition of those sequence M create incompatibility between the differences and the similarities. And you have the sequences M, which is associated with the sequence in the relative aspects. Then G of M has two components, each require two recombinations, but this combined networks needs only three models to fit in. So from four, it can be reduced to three, and the additions can be understood. The practical sufficient condition is if M can be determined from the network N for every edge recombination at one site, then every node is labeled with the sequence M, then there is a fully decomposed network M, which minimizes the number of recombination over all possible networks M. This is also an associative property, the practical sufficient condition. Another practical sufficient condition is the M can be determined from the network N, where the number of recombination equal to the polycomputable haplotype for a lower bound. Then there is a fully decomposed network for M, which minimizes the number of recombination over all possible networks. What's a perfect phylogeny for a non-binary character? Still now we are talking about binary. So the input consists of n sequences for the matrix M with m sides of characters, where each side can take the k states. In a perfect phylogeny, T for m, each node of T is labeled with m length of sequence, where each side has a value of range 1 to k. So T has n leaves. So one of the each sequence in M is labeled to that sequence. So in other case, for each character state pair, that is C supported with a small s for the node T, the labeling can be from a connected tree. And then a perfect phylogeny is associated with the input. You can see the transformation matrix here. You can even call it as transition probability matrix. And you can see the tree for state 2 of character B is being analyzed separately. And then we have the caudal graph. The basic definition is graph G is caudal if every cycle of length 4 or more contains caudal, and A, a graph G is caudal if and only if every minimal vertex separator in graph G is a click, because in a graph for identifying the hubs, you have various centrality measures. So every centrality measure will be giving you a relative hub. So by using most of the centrality measure, which hub is being generated, we say it's a click, and the relative hub gives you relative clicks. So based on the maximum support where a hub gets is the specific click we need to select, so that only the maximum click centrality is one of the technique people are using now. The caudal graph have large number of applications, and they are more based on the separator results with respect to the basic definition. Caudal graph on n nodes can have most of the n maximal clicks. Probability basics only. Another classic caudal graph theorem gives for each node the graph is a member of S. That is the set of sequence. Another classic caudal graph theorem, what it says is that a graph G is caudal if and only if the intersection of the graph of A with the set of substrate tree, and each graph is a member of S associative property. Then we can relate the graph to the perfect phylogeny. A perfect phylogeny, T is associated with the table for the character C if and only if the subforest of the T is induced by the set of node labels C and X to form a single connected subtree of T. So there is a natural set of subtrees T induced by E, where E is the complete table. So the caudal completion approach gives you a perfect phylogeny also, where a graph G of E has one node for each character state pair in E and the edge between two nodes, if and only if there is a row in E with both these character state pairs. So when it go a little deeper, C of E be the graph derived from G dash of E as follows a node in the C of E for each maximal click G dash of E. So as I told you, in a protein-protein interaction network, you can have a multiple click but which click is supported by the maximum centrality measures is the one you need to consider for the analysis. So you can create a edge UV in the node CE where a minimum of clicks for U and V is associated with the graph G dash of E share one node. So these are the mathematical conclusions they bring in the graph theory. So for the weighted edge UV, we can have more number of shared nodes. So the C of E can be created from G dash of E in polynomial time. So any maximum spinning tree T in which the C of E is a perfect phylogeny, for E you have association with G dash of E. So in case of the perfect phylogeny, this problem is being studied for 20 years. Little solutions were given by various people. For a fixed bound, the number of states per character have association to determine the perfect phylogeny problem to be solved in polynomial time. However, the number of states per character is not completely bounded and it's tough to determine also. Then we come to the NP complete problem. So this is the small example to give you the subset of the adjacency matrix associated with the graph theory. So this concludes my presentation, and you are very welcome to give your queries, and I am here to answer your queries.
Thank you, sir.
Thanks a lot.
Thank you, sir, for your wonderful presentation. It has been very informative for our participants. Now it's time for our quiz session. After completed quiz session, we come back again in our question answer session. Okay, sir. Hello, sir.
Okay, fine. Yeah.
Okay. Dear participant, it's time for our quiz session. After completed our quiz session, come back in our question answer session. It's time for our quiz session. To join our quiz session, you have to go www.slido.com. Then you have to enter the code, and the code is IGP3, and the code is IGP3. After short video, we will start our quiz session. In the meantime, you can join in our quiz session, and Dear participant, to join our quiz session, you have to go www.slido.com, then you have to enter the code, and the code is IGP3. We encourage everyone to participate in our live quiz competition. Just go to directly www.slido.com, then you have to enter the code, and the code is IGP3. And top 10 winners will be awarded with quiz competition certificates. First question: what is artificial intelligence? Is it program with your own intelligence? Second option: making machine intelligent. And the correct answer is option 2, making a machine intelligent. Now we go for our second now question. And the second question is, in which university the first demonstration of AI program ran? Option 1, Cambridge Mellon University. Second option, Oxford University. And the correct answer is option 1, Cambridge Mellon University. And 43% people are right. Our third question is, machine learning takes data and looks for dash trends. Option 1, underlining. Second option, famous. And the correct answer is underlying, and 74% people are right. Now we go for our fourth now question, and the question is, AI generally is undertaken in conjunction with machine learning and data. Is it option 1, analytic, 7 option, demonstration? And the correct answer is option 1, analytic, and 97% people are right. Um, Fifth now question, what are the two types of artificial intelligence? Is it option 1, narrow and general, second option, specific and general. And the correct answer is narrow and general, and 23% people are right. Now I go for our sixth one question. Dash is the goal of artificial intelligence. Option one to extract scientific process. Second option to explain various sorts of intelligence. Dash is the goal of artificial intelligence. Option is it option one or option two? And the correct answer is option two to explain various sorts of intelligence. And 74% people are right. Now we go for our seventh one question. Now we go for our seventh one question. And the question is stores information in the form of logically related to dimensional tables. First option, data model. Second option, rational database model. Now you can see our correct answer, and the correct answer is option 2, relational database. So, our eighth now question is Agoda website use application programming interface API option 1 or option 2. And and the correct answer is true. Our ninth one question: artificial intelligence, the content people see in their social feeds and what ads are shown to them. Is it true or false? And the correct answer is true, and 95% people are right. And now we go for our last and final question. And the question is GOR paid robotics, artificial intelligence such as chatbot are sample of latest technology. Is it true or false? Mm, And the correct answer is true, and 100% people are right. And our quiz competition is complete, and we can see our top ranker. And the top ranker is Kidwana January CMB. Then we have Oji, then we have Corajan, then we have Safal, then we have Joel Apricot Barnaser. Congratulations, our top 10 quiz competition. We have all of your name which you entered during joining. After completed our webinar, we posted quiz competition winners, and top 10 will be awarded with quiz competition certificates. Our Facebook official page is Institution of Global Professional. Participants are requested to ask their question in our comment box. Please address speaker name, then ask your question. After short video, we'll start our question answer session. So let's enjoy our question answer session. Participate. Our speaker have some technical problem for that he can't join with us. We have to wait for some minutes. Uh, so we have wait for some minutes. If he come in our question answer session, we Dear participant, our speaker have faced some technical issue. For that he can't join with us. So we have to wait for some minutes our speaker. If he is come, we continue our question answer session. If he can't join with us, we give you a webinar code. So dear participant, please wait for some minutes. Dear participant, we can't connect our speaker. Uh, for that, we can't complete our question answer session. It's time for our to give you our webinar code. Our webinar code is IGP1634. The code is IGP1634. Without code, no one is eligible. With code, you can claim your certificate anytime. And the code is IGP1634. Thank you everyone for your active participation. See you in our next session. Till then, stay happy and blessed. See you again our next session. Till then, stay happy and blessed. Thank you so much everyone. Once again, I am Yoni Sakta from Bangladesh.